Welcome to Guide Guru. We hope this video helps.
You begin by creating a new project and importing your raw data so you can work immediately. Once your files are in, assign a reference sequence to guide alignments. This improves downstream mapping accuracy. Make sure to check import file types, FASTA, FAST, ABI, and confirm sequence encoding. Then, organize sequences into folders and apply clear names. Doing this now avoids confusion later and speeds analysis, because Geneious uses project context to remember settings and speed up repetitive tasks. You now align sequences using MAFT or muscle. Choose gap penalties and scoring to match your data. After alignment, inspect the alignment view, zoom into problem regions, and trim low-quality ends or ambiguous bases. This improves consensus accuracy. If assembling reads, run the de novo or reference mapper and review contigs for coverage. Once done, generate a consensus sequence or call variants. These precise edits ensure your final sequences reflect biological reality, not sequencing artifacts. You finish by adding annotations, genes, coding sequences, primer sites, so results are interpretable and reusable. Then export in the format you need: GenBank for submission, FASTA for phylogenetics, BAM or VCF for mapped reads. Make sure metadata and feature keys are correct before export. If something goes wrong, first verify file types and import settings, then check memory allocation and rerun alignment with adjusted parameters. This troubleshooting step usually resolves common errors quickly.
Thanks for watching. Consider subscribing if we helped you today.